Good morning. My name is Tess Danielson. I'm an environmental protection specialist for DOEE, and I work in the monitoring and assessment branch in the water quality division. So today I'm going to give you an introduction to our phytoplankton monitoring program, as well as go over some of the lab methods that we use for phytoplankton analysis. So before we get started, I'm going to minimize my video here so you all can see the slides. All right. Next. There we go. Okay, so why do we monitor phytoplankton in the district? Well, there's a couple of reasons for that. First is the role phytoplankton plays in water quality. It can act as a bioindicator of a healthy system. For example, a healthy system would have a nutrient concentration that supports phytoplankton growth. If there are too many nutrients or eutrophication, that can lead to blooms that can negatively impact the water quality. For example, can limit sunlight and deplete oxygen as well. Speaking of which, phytoplankton are oxygen producers. They are primary producers in the base of the aquatic food web. So they're an important part of population dynamics in the water. And so they also support dissolved oxygen levels. And identifying the taxonomy and density of phytoplankton present in major water bodies can share important ecological dynamics as well. So the third reason we monitor phytoplankton is for public safety. As I mentioned, blooms can occur that can be harmful, and that is something we want to monitor. So our monitoring program occurs on both the rivers in DC, the Anacostia and Potomac, as well as tributaries that aren't mapped here. So DOEE analyzes two sites, the PMS 10 here on the Potomac and ANA 01 on the Anacostia. And the other sites listed here, as well as tributary sites, are sent to contractors for analysis. We're alerted of any blooms in the district through citizens or other agencies. And when these events occur, we test for toxins and also do cyanobacteria counts to see the levels of cyanobacteria and toxins during that specific event. And there are toxin thresholds that are used to help guide what levels are safe for recreation. And when these counts or toxin levels surpass this threshold, we notify the public either by signs or public memos there, so everyone can stay safe. Chlorophyll A is also sampled at all of these sites using a YSI sonde, an optical sensor that is attached. And this is used as a proxy measurement of algae. So here are two examples of harmful algal blooms in the district. The first is the picture on the left here is of a bloom in the Potomac River that was dominated by Pseudoanabana. And you can see here the microscopic view of these filaments. So this species here produces microcystins, which is a liver toxin. And over here on the right is a bloom that occurred in 2019 that was dominated by Sphaerospermopsis. And this is in the Constitution Gardens Lake on the mall. And you can see here, it's a little faint, but this is a picture of the Spermopsis under microscope. And this species produces an anatoxin A, which is a nerve toxin, and is dangerous because it can impair synapses and inhibit enzyme activity in the cells and in the nerves. Excuse me. So not all these blooms are harmful. Certain species, as I mentioned here, can produce different toxins, and at high levels, they can cause bodily harm when ingested or inhaled from aspirated droplets. So in the case of the Constitution Gardens here, the levels of these toxins and the counts of the cyanobacteria did not surpass the threshold. However, we did have, signs and warnings not to let pets or small children in the water here, just as a precaution. All right, so this is a diagram showing the drivers of blooms, specifically in the Potomac River. Actually, this was from a paper, a local research paper. And you can see the different parameters here that can lead to blooms: flow, water temperature, mixing, sunlight, and salinity. There's also research available for the Anacostia River that links phytoplankton composition to flow and nutrient concentration. These things can be correlated because different phytoplankton have different preferences for nitrogen forms to uptake for their physiological processes. So cyanobacteria tend to bloom in polluted conditions or when nutrients, the water is over-enriched with nutrients. And this is because they prefer ammonia and urea nitrogen forms for uptake compared to diatoms, which are more prevalent when nitrate is the dominant nitrogen form. So those are just some examples of local research to harmful algal blooms in the DC area. All right, so I'm going to move on here to our methods of collection and preservation. So let's see here. Let me pull up these videos that I took of our collection techniques. And here. Nope. Screen. Here we go. All right, here's the first video. The sound will come through. You're collecting phytoplankton. Oh. Grab samples, so you take your bottle, dip it under the surface, all the way up to the top, leave a little room to add the preservatives. Okay, so hopefully the sound came through clear, but basically we're just doing a grab sample to collect the water and the phytoplankton within it. So here's the next video of the preservation. All right, once you've returned to the lab with your sample collection, you want to add 5 mL of Lugol solution, which will preserve the phytoplankton for later collection. And you can see here, as it dispenses, it's this amber color. You can sort of see it. So that's how you know also when you open the bottle later in the lab to check that it has been added. You'll see that amber yellow color in the bottle. And then you want to store this at room temperature in this dark opaque bottle until future identification. Okay, so we add the preservative Lugols there, and it can be stored in the lab for later processing. Okay, so let's go back to the screen here. Next. All right. So I'm going to give some background on taxonomy of phytoplankton. A quick note here is that there are two major kingdoms, prokaryotic algae, which the cyanobacteria fall into this kingdom, and eukaryotic algae, which all the other classes fall under. So the main difference here is that cyanobacteria don't have organelles, and eukaryotic algae do. They have the nucleus and chloroplasts and mitochondria and everything else. All right. So there are some major eukaryotic algae groups here, and they can be classified based on color and pigments. So you have chlorophyta, which are green algae, phaeophyta, which are brown algae, pyrophyta, which are the dinoflagellates, chrysophyta are the diatoms, rhodophyta is red, and euglenophyta are euglenoids. So these are the major divisions or phylum here, and we see most of these different phylum in the Potomac and Anacostia rivers. Oh, and one more note here, for our purposes, we try and identify the plankton to at least the phylum and then down to the lowest taxonomic level, which is usually the genus. All right. So to prepare for the identification and enumeration of these phytoplankton in the lab, there are a few steps to do before we start that. So one of these steps includes settling the samples to concentrate it. So in some months, there's not a high concentration, or sometimes time of day, there's not a high concentration of phytoplankton in the water. So we'll just use gravity settling method to concentrate these samples, so they're easier to count all in one go. So once this happens, you have to wait. Usually if you're settling 50 mils, you have to wait about 50 hours, an hour for each mil. And once this happens, you decant the sample and keep whatever concentration you're going for and store it in a small vial. And I will probably go over this in more detail using a video in a different presentation. So it should be more clear then. So once you have the sample, it's best to identify which species is which at a higher magnification first, because usually our method for enumeration, the magnification on the microscope only works at a certain magnification for the type of slide we use to enumerate. So first we put it on a regular slide, microscope slide, and ID, so we can get a closer look and an idea of what kind of species we're looking at in that sample. So there's also a camera attached to the microscope that we use to take reference pictures for plankton that are harder to identify or that you haven't come across before. So you can use some keys to look up those species later. So here is just a picture of what you're looking at under the microscope. And this is this is at I think the 10x objective. So it's not very magnified. So you can see species are there, but you want to get a closer look at most of these. So here's some closer looks, some examples. of microscope of phytoplankton we see in the Anacostia and Potomac rivers and their classification here. Just a quick note, species classification is kind of always in review and changing, and the categorization does change, or they're given new orders or families are created to fit a certain species as research continues. Generally though, the genus and for the species we're looking at stays the same. So here is an example in the division Pyrophyta. And so that's a dinoflagellate, and we're looking at the genus Gymnodium here. Another example here of the phylum Euglenozoa. And so here we're looking at a euglenoid. And this last example here is a chlorophyta, which is pretty common in district waters. And this one is Cenedesmus. So you can see the different structure here compared to the others. It has these spines on them and this like 4-layered ovals there. So those are just some examples. There's a lot more you see in these rivers, and hopefully we can do another presentation where I give you a closer look and you can practice some identification. The other half of this analysis is the enumeration. So we not only identify, we want to count how many of each of these organisms we see. So with your sample is in the small vial, you want to agitate that, shake it up so you get a good mix. You're not just pulling from one part, you want it really homogenized. And you draw 1 milliliter from that vial and pour that into a counting chamber, which is called a Sedgwick rafter cell. So you can see here, there's kind of a dip in it. So it actually creates a volume of there. So once, oh, I skipped a step here. So when you're counting, if you were to count this whole chamber across, that would give you cells per milliliter. And that would take a long time. So there's some formulas used to extrapolate how many cells per the whole chamber here. And I'll get to that next. But before you start counting, you want to allow the phytoplankton to settle for 15 minutes. And then you have this Whipple grid, which is it's in the ocular piece of the microscope. So when you're looking down into the microscope, you see this little grid here, which is helpful for keeping track of where you're going as you're counting along the slide here. And again, I'll go over this in the next presentation with some video, so it'll be easier to understand. But as you're counting, you want to get a minimum of 200 cells, and you're only counting the live and unbroken cells. And live is a bit of a misnomer here because they aren't alive anymore. But any kind of cells that are broken or just don't have any color to them anymore usually means they were dead before collection. So you're not counting those in your totals. All right, so we're going to skip ahead to the identification notes here. So as you can see, as you're counting, you want to take some detailed notes and even little sketches. And the reference image, what I mentioned before, you can use the camera to take a picture of these images or of the species, so you can refer back to them later. But yeah, little notes, you want to also note the cell size, so you can confirm, that genus or species falls into that size category. Just another way to help you identify them. So to get the final count of cells per milliliter, we use this formula here of the number of cells counted, the area of the Whipple grid or of the field, which is the Whipple grid image area, the depth of the field of the subject raptor cell depth, and the number of fields counted. So most of these numbers are fixed when you're counting at a 10x objective. The area is 0.25 millimeters squared, the depth is 1 millimeter, and if you count just one strip across, you slide the subject raptor cell all the way across, counting as you go, then that would just be one field. If you don't get to 200, if you don't get your count up to 200 in that one field, you continue on to the next. So then it would be 2 fields. So then you have a whole number here, and cells per milliliter would be the number of cells you count times 40. And then that is divided by the concentration factor or multiplied by the dilution factor if you dilute or concentrate the sample. All right, so here is an example of the finalized data. And this data can be used in a number of ways. For example, you could analyze it to determine the percent contribution of each phylum. So like how many diatoms or how many chlorophyta are in this water sample. And you could also use the other water quality parameters taken during when this sample was collected to correlate or find other trends. So this one is an example of the Potomac River when the chlorophyll was 3.2 micrograms per liter. This was back in 2018. So you can see the total cell density here was 2,190 cells per milliliter, excuse me. And an example of the Anacostia River is from February of 2020. You can see the chlorophyll was at 2.17 micrograms per liter. And you have a similar cell density here of 2,848. But the contribution of different phytoplankton differs between these two rivers here. You can see a little more of the chlorophyta, the green algae here, compared to maybe more of the diatoms, and a little more diversity here in the Potomac River sample. All right, so some key resources here that I use a lot to help identify the different plankton are these FICO keys, and the Phycotech SmugMug account is really helpful as well. They're all like photo accounts, and I can show you those links, but they're sorted by phylum and then also morphological type. So I have this slide here I borrowed of morphological type examples. So you can see unicells, which is a singular cell, filaments, which are kind of how they sound, longer strands. They can be branching here or not branching here. Colonies of cells can be small, large. They can be kind of defined in or distinctive shape versus amorphous here. And obviously there's microscopic algae versus macroscopic, which we don't deal with in our analysis. We're just focusing on microscopic phytoplankton. So let me share with you these websites. Okay, well, there's one here. I don't know if you can see this. No, you cannot. All right. So one example here is, and there's actually a few of these booklets in the lab at UDC that we can hand out or I can have available there. This is a USGS guide to freshwater cyanobacteria. So this booklet shows some, too fast, some common types. Okay. And has a lot of pictures to share and other background information on cyanobacteria. The other window is on this page. Nope. Just a second. Okay, well, let me. Recording in progress. Okay, we got it. Here is the PhytoKey website. So it's very helpful. You can just click based on image here. So if you see something kind of squishy and long, you're probably looking at a euglenoid, and you can click further to see different species and various photos of them. So PhytoKey is pretty helpful with that. If you don't really know what you're looking at yet. The Phycotech website is also very helpful and has a lot of variety in their photos. If you do know more of the division of algae you're looking at, then you can click there. So if I'm looking at a green algae, I'd click here and scroll through the images. If I know it's a filament, I want to keep scrolling till I see something that looks like what I'm looking at. Oh, it kind of looks like this. I'll click here to see more images and get a closer look. So this site's very useful as well. And nope. The last screen here is AlgaeBase, which is a global algal database of taxonomic nomenclature and distributional information. So for example, if you want to more in depth about a certain species or genus like Volvax, you can type that in there. You'll get the taxonomy information here, and then a bunch of references for and description about it. And this nomenclature notes here, I was mentioning before that the names often change, the scientific names. So it'll give you previous names, recent taxonomic treatment, all that sort of stuff. So another helpful site. All right, so I invite you to go visit those sites and familiarize yourself with those resources, because I believe next time I will do the presentation, I'll show you more about the lab methods and then also show you some slides up close, and we can have you try and identify some of these species. So I want to thank you for listening and taking the time. And if you have any questions, please pass them on through email. And thanks again.